Today it is my pleasure to introduce Brian Smith, and no doubt most of you know him. He received his Bachelor of Science at Juniata College in Pennsylvania and then went on to get his PhD in entomology at the University of Kansas. He did several postdocs, went to Ohio State, arrived here at ASU in 2005, and currently has funding from NSF, NIH, and other agencies. He is a behavioral neuroscientist and is interested in learning and memory in insects and mammals, and also how animals learn about odors. And today he's going to tell us about the roles of social networks in disease transmission and individual decision making using the honeybee as a model.
Well, thank you very much. It's a real pleasure to come here and talk about a lot of the work my labs have been involved in for many years now. And as Ann said, a lot of it's been learning and memory, using this animal right here, the honeybee, as a model for understanding, well, how do these animals with such small brains manage to learn so much about their environment and perform feats of learning and memory that most people would find impressive for such an animal with a small brain. And I'll talk a little bit about that work today. I decided I would go into that because I'm going to talk about this little brain here and the reward system in the brain, the reinforcement system, the reinforcement pathways. And you're familiar about that with mammalian brains, dopamine and various other things that represent something good that just happened, and we should learn about it because we want to identify that good thing in the future. Honeybees have similar kinds or similar functioning reward pathways in their brains. And I'm going to tell you about those pathways and how they work into decision making, in part because I think it's fascinating, and also in part because a lot of what I want to tell you down the road about how these pathways are involved in decisions that groups of honeybees make, and how this may be important for understanding how patterns of disease can flow across social groups. These are areas that my lab has been involved in over the years. This is a newly emerging one that we're getting involved in, and I'll tell you a bit about that. This is one that we did several years ago, or I did with a postdoctoral researcher then in my lab. And I'm not going to be able to tell you about some of the work, giving a little bit of a shout out to these other animals, mammals and fruit flies. I have a whole menagerie in my lab now. And because they're very good for genetic manipulations and various kinds of things you can do, they turn out to be wonderful animal models. And I also wanted to give some recognition to the animals I got started with some 30 years ago at the University of Kansas for my PhD thesis, because a lot of what I tell you really stemmed from the work I got started back then. But one of the reasons I wanted to point out that these are important, of course, honeybees are wonderful in terms of the social networks and what we understand about the social networks in the colony and how we can experimentally manipulate those networks to test hypotheses. But these other animals, this is a primitively social bee, Lasioglossum zephyrum, I did my PhD thesis on. They're also very social, much smaller colonies, and they differ from honeybees in certain ways. So when a forager sweat bee here finds a flower and comes back to the colony, it forages on its own. It doesn't recruit other bees in the colony as far as anybody is known. Honeybees actively recruit other workers to sites they've found. So that's a big difference between these two kinds of bees. They don't engage in a kind of food exchange called trophallaxis. When they come back, they don't turn around and feed the other bees in the colony. And you'll see as I get on in my talk why this lack of recruitment and lack of trophallaxis is important, because everything I tell you about bees will depend on those two things at the end of my talk, and at least I want you to walk out at the end of my talk knowing there are some fascinating animals out there for which everything I said could be very different and worth looking at. Let's start off with a honeybee brain. That tiny brain has about 1 million neurons, about 1/10,000th the size of our brains. And the brain is very plastic. The neural networks in various locations of the brain you'll see, can be changed as the animal learns the association of a pattern of neural activity with something it wants to get, sugar, nectar, sugar water in the case of reinforcement in our studies. Several years ago, when I was doing one of my first postdocs with Randolph Menzel in Berlin, Germany, they had just identified a series of neurons in the brain. This is one of them. They're called ventral unpaired medial cells, because if you take this as the brain and look at it in schematic here, the cell body sits here at the base of the brain, sort of behind and in back of the brain, and it's in a center that receives sensory information from sucrose-sensitive taste receptors on the bee's mouth parts. So when the bee's feeding on sugar, this cell gets a lot of input, and the outputs you see come up and arborize extensively all over the brain, in this area that I'll talk about in a minute called the antennal lobes, as well as into an area up here called the mushroom bodies. And the mushroom bodies alone, that's about a third of the brain. They're highly multimodal. It's where the bees actually make complex associations in their environment, and when they're sleeping at night, I think they probably compose poetry with these neuropills. They're very complicated, fascinating things. But this is the classic kind of a distribution of a modulatory neuron. And at least now the model, and there's a lot of evidence for it, is that whenever the bee feeds on sucrose, this neuron literally releases a biogenic amine called octopamine all over the brain. And when octopamine elevates, that's a signal to remember whatever just happened, because that's associated with something important. It's associated with food. Now, something I became interested in as sort of my focus for so many years is this area, the antennal lobes, because the neurons here on the antennae, there are about 50,000 neurons on each antennae that are olfactory sensory neurons. They pick up odors, floral odors. And they transmit their axons here to the base of the brain, which is the antennal lobe, where they make the first synaptic contact with interneurons in the brain. And I became very interested in knowing, because a lot of people would talk about this area of the brain, both in mammals, which is the olfactory bulb, and in insects, as just sort of a summation of sensory activity in the sensory epithelium, which is the nose or the antenna. And I began to wonder, well, if that's really true, why would the antennal lobe need to know when reinforcement is present, unless it's driving some kind of plasticity? And I'll tell you about those studies and how it does that as I get into the last part of my talk. First, what about some of the pathways? We talked to you about octopamine, and this is a slide made by Irina Sinikiewicz, sitting right here in my lab. These are antibodies, and I'll focus mostly right here, but if you do a cross-section of the antennal lobe, you would see these little grape-like clusters, and I'm not doing them justice by describing them that way, but they're clusters of synaptic contact with sensory neurons, and they're called glomeruli. And if you take a cross-section of a glomerulus, here you'll see that VUM neuron, the processes of that VUM neuron with an anti-octopamine antibody that's doing something to modulate the first contact of those sensory axons with the kinds of local interneurons and projection neurons that also innervate this area of the brain. There are other serotonin, other biogenic amines, dopamine, ones that you may be a little bit more familiar with because they're also found in humans. So this neuropill is a very rich it's very rich in plasticity, let's say, that can be modulated by release of these biogenic amines that could be doing some very different things. Well, a nice thing about bees is you can bring them in the laboratory and ask them questions. This is the way a bee frequently appears in my lab. We strap them down into a little harness. It's a metal device here. It can be as sophisticated as a section of a soda straw from McDonald's, which we parasitize every once in a while when we need bee harnesses. It's the only reason I go to McDonald's, the only reason to go to McDonald's. But you put them in this harness and they can freely move their antennae. That's their noses. I just told you the 50,000 sensory receptors project into the brain from the antennae. And when we blow an odor across the antennae, the bee frequently doesn't extend its proboscis right here. But after you feed the bee sucrose a couple of times, so you give them just the odor, they'll extend their proboscis as a conditioned reflex. So it's classical Pavlovian conditioning. The odor comes on. At first, there's no response, and you feed sucrose, and then after 1 or 2 or 3 trials, you get a nice conditioned response. It's our way of asking a bee, can they detect an odor, and can they discriminate it? This is what some conditioning trials would look like if you give them a very easy task. This odor is associated with sucrose. Very few bees respond on the first trial. So this is a % response across a population of about 30 bees that we were training. Very few respond, but after a single reinforcement of that odor with sucrose, suddenly 80% on the second trial, and then it jumps up to frequently close to all the bees responding after a few trials. It's very easy to train bees. By the way, this fast learning about odors is not peculiar to bees. Mammals show it too. So the mice in our laboratory, if you give them such a simple task as this, they'll pick it up very fast. And then once you train them to this odor, we test them to that odor, they all respond, and we start changing the structural features of the odor to be more dissimilar from the conditioned odor, and you see these nice generalization curves, meaning that there's some way of mapping these molecular features of odors onto the representation in the bee's brain. Of course, we can make the conditioning very difficult and ask them to discriminate 2 odors that are very similar. And this is a case here, and you'll see they're not going from 10 to 100 really fast. They're going 10, 60, 80, and they're hovering around here. And then this is an odor that's not associated with sucrose. And they start to generalize at first after they get this reward, but they never really increase to this odor like they do to this one. And this is an even harder task. Here's the conditioned odor. It's going up in response, and they're not really discriminating it from the unreinforced odor at first. But I got in trouble with some reviewers because I said this is the aha effect. Sooner or later, the bees act as though they're thinking, if they can think, okay, I get it. This one's reinforced, this one's not, and you'll see them diverge. That's classical, again, you see that in vertebrates as well. And then afterwards, you can establish these nice generalization gradients depending on whether it's the CS plus and minus or CS plus here and minus here. So it's the basis for a lot of the physiology I'm going to show you is this kind of conditioning. But several years ago, and this is going to be important for some of the group-driven behavior that I talk about, we started studying another kind of learning. It's called latent inhibition. And most of you right now in this room could be paying attention to 1,000 different stimuli. Hopefully, most of you are paying attention to me, right? So there are things that are happening here that you're not paying attention to at all. And you're learning, actually, that they're not important. And it's a very important kind of learning. It's learning to ignore things. Because if you can't ignore things, you can't focus on the right things to pay attention to. It's ubiquitous in animals. Every animal shows it. Humans show it. Schizophrenics are supposedly deficient in their ability, ability to ignore things, so they're getting bombarded with stimuli and have a difficult time picking out the right stimuli to pay attention to. And I just saw a wonderful talk at a scientific meeting last week about how a small roundworm with something like a whopping 308 neurons in its entire central nervous system shows latent inhibition. So what is it? We're going to give them an odor for anywhere from 0 to 50 trials. Usually after about 30 or 40 trials, what we'll do is we'll turn around and train that odor associated with sucrose in exactly the way I just showed you. So they should learn it, right? And in parallel, on different trials, they'll get a different odor also paired with sucrose. So they should learn these 2 odors equally, and indeed they would under normal circumstances. But what happens is that odor that wasn't pre-exposed, they learn it well. The one that was pre-exposed, they start to learn it, but they never quite learn it as well because they've just learned to ignore it, and I'm just teaching them something different, and they have to overcome that learned ignorance, if you will. Okay? This is latent inhibition, the difference between these two curves. Now, another talk is this difference depends on the odor independent of the visual context. The differences down here are very much dependent on an association with a visual context. So it's a context. It's not as simple as it looks. It can be context driven. But I'm going to focus on part of my talk for latent inhibition. So now, what about the reward pathways in the brain? And we use this antennal lobe as sort of a model, but remember what I'm going to tell you about the plasticity here is happening in parallel all over the brain, driven by that VUM neuron and the octopamine that it releases. One thing we can do, and we learned this from a close colleague, Giovanni Galizia at the University of Konstanz in Germany, is here's the antennal lobe of a bee brain. Here's one of the axon tracts of the projection neurons carrying information out of the antennal lobe. When we create a small lesion right here with a dye called Fura, Fura2, was isolated from jellyfish, and it's the dye that caused jellyfish to fluoresce. If you've ever been at the ocean and you've seen a wave come in and these jellyfish will start to light up, that's Fura. It was isolated from jellyfish many years ago. And this dye will retrogradely fill along this axon tract and fill the dendrites of some of these projection neurons on the dorsal surface of the antennal lobe. So, in terms of a mapping, what we're going to do is look at this antennal lobe at the dorsal surface and the neural activity as we stimulate a bee with an odor. And if I project this onto what you would see looking through the microscope, it would look very much like that. Now, once we stimulate with an odor, imagine you can superimpose this onto here, and you can see when the odor comes on, a little red square right there, you'll see Fura will start to emit light at one wavelength, absorb light at another. If we take the ratio of the two, we can get a good activity of neural activity, because what it's doing is tracking the elevation of calcium. I forgot to say that. When the neurons become activated, calcium transiently increases and then is rapidly buffered back to a baseline level. And when calcium increases, it reacts with Fura to cause the emission and absorption of light at different wavelengths. So we can measure neural activity. So what does that activity look like to different odors? You were looking at maybe a second's worth of activity as we stimulate with an odor. So imagine we're just taking a snapshot over that second and averaging all of those frames together. What you would see to one odor, hexanol, you'd see a spatial pattern, a particular spatial pattern across all of those glomeruli. If you look at a different odor, octanone, you see there's a different spatial pattern, and that's the basis for the bee being able to discriminate the neural representations of these two odors. But if we start mixing them, 9 parts one to 1 part of the other, 7, 3, 5, 5, 3, 7, 1, 9, so we're grading these stimuli into one another, you'll see that you don't get, it's not like it's all the same and then suddenly you get a nonlinear shift. It's a pretty smooth gradation of the pattern from one extreme to the other. Moreover, if we now, instead of averaging all the frames together, we perform a statistical analysis, a principal components analysis across all the 20 different areas we're measuring and across time, we can represent, using 2 of these statistical factors, the activity to each of these odors. So this is one pure odor, and each of these little X's that you may or may not be able to see is a time slice of about 100 milliseconds. And you can see the odor comes on. This whole, if I call them transients, that's what my colleagues, my mathematical colleagues call them, transients, because the activity evolves out to a peak, and then when the odor comes off, it comes back to baseline. This is the other odor. And you can see when we shift from 10-0-9-1-7-3-5-5-3-7-1-9-0-10. It's a nice smooth gradation across those different odors. And that's a perceptual dimension for this dimension I'm manipulating, and it's represented in the brain. And that's not trivial because, like I said, a lot of people would predict that you would see a lot of these transients clustering over here, and then when you get to 5-5, it performs a nonlinear shift and goes over there. So it's not trivial to try to configure a neural network to make a smooth transition like that, but the bees do it. And I won't go into that, but now I want to talk about plasticity. If you remember that octopamine neuron that's raining down octopamine all over the brain when something good happens, in this case when the bee gets a little bit of food. So we take naive bees, we're making making these transients, and this is now 9-1-1-9, and all the different mixtures in between, and that's what I just showed you. But we've also taken a group of bees and we've told them that 9-1, this pattern, means sugar, means food, it's good. We've also told them that this 1-9, the opposite end of the spectrum, without getting to the pure odor, means nothing. It's bad, so discriminated. This is the basis for that behavioral discrimination I showed you some slides ago. It's now looking at the neural representation and what happens. So we can calculate for each of these particular time points in 20 different dimensions the Euclidean distance between these transients that represent these two odors. And we can plot the distance at each time step. And what you'll see for the naive bees is the odor comes on about right here. The transients start to separate. They reach a peak separation, and then they gradually come back to baseline when the odor goes off. If we've trained them, however, that this is good and this is bad, or this is good and this is nothing, then what happens is they get farther apart. And what happens is basically what we've done is we've taken that perceptual dimension, which is relevant to the bees right here, right now, today, and we said it's important, and what the neural networks are doing is it's stretching that dimension to help the bees make fine discriminations along it. That can go away tomorrow if the program changes, and that's the importance of plasticity. So that's one of the things that this octopamine is doing in the brain. We've done quite a bit of computational modeling. I mean my colleagues have. I don't do the math. And we set up these neural networks based on what we think is a pretty reasonable approximation of the connectivities. And one thing that's come out in several efforts is that there's a most reliable way to modify these networks to produce the results that we see right here, and that's you target the inhibition. In other words, you could target the octopamine neuron, you could target the excitatory axons coming in, you could target the excitation going out, but there are local interconnections that are inhibitory, and that's the easiest way to do that. And Arena first tested this several years ago when we developed an antibody against a receptor for octopamine. That is, cells that express this receptor are the ones listening to that octopamine reinforcement signal. And it turns out these are the cells that express the receptor. These are GABAergic inhibitory interneurons in the same area of the glomerulus. Overlay these two images, and wherever you see white, those are GABAergic local interneurons that express the octopamine receptor. So that was a wonderful test of a lot of the predictions from the computational model. So we're starting to know a lot more about how plasticity occurs in the brain. And what I just told you for the antennal lobe, by and large, as far as we know, also occurs in those mushroom bodies, the big poetry composing areas. You see GABAergic interneurons expressing octopamine receptors there. So we think what we're looking at is something general about how you drive these networks using plasticity. Now I'm going to skim over this part because otherwise I'll run out of time. or is everybody free until 4 today, which is how long it's going to take me to get through this if I don't skim? Floral odors are much more complex than the kinds of odors we use in the lab. And in fact, if you take 2 different varieties of the same species of flower, you sample the floral odors, as we and our colleagues did several years ago for snapdragon, for example, there may be 6 or 8 or 10 different components. And if you start looking from flower to flower, you'll see some variability. So one flower is not exactly like the next one, and neither is exactly like the third one. And the separation between the 2 varieties is really more statistical than anything else, based on the mixture and not 3 new components over here that weren't over here. So it's a very fine discrimination. So if you plot the flowers of one variety, you get a cloud of points here, these orange points, and if you plot a bunch of flowers from a different variety, you get a cloud of points over here. So this is the problem for the bee to make that discrimination. They do it very well, and I'm just going to go through this quickly so I can get to the rest of my talk. And these are 6 of the different components that are in these complicated mixtures. The bees here can, and we did this analysis to show simply that the bees can perceive all of the components. So this is the response across 20 areas of the antennal lobe that we measure, and you'll see they all pretty much show responses to each of the components of this flower. And now the problem is, how do the bees separate those clouds of points? And this problem, just sort of as a brief aside, where I said a bee lands on a flower and it gets reinforcement. Normally, the way we think about learning is the animal then has to find the exact same thing to predict reward in the next place at some future time. And even Pavlov, 100 years ago, recognized that's not the problem, because he said every stimulus differs from one time to the next, even under the most highly controlled laboratory circumstances. So in this case, if the bee said, I got to find exactly that same odor, the only thing out there that has exactly that same odor is the same flower it just depleted of nectar. So it's kind of a stupid thing to do, not a smart thing to do. So the problem for the bee is not, is this the same, but is this similar enough that I'm going to cluster them together, right? And I'm going to say this odor is similar enough to mean something the same, or it's too dissimilar, so it must mean something different. Can the antennal lobe help to do that? And it can. So these are just some artificial mixtures we made up to sort of simulate the variance across those clouds. And just to get to the point here, if you look at the correlation of neural responses to each of those 6 artificial mixtures within the green variety or within that orange variety, the correlations of neural responses from one mixture to the next are pretty high. They're on the order of about 0.9 if you can't read these values. This is the mean here, a correlation within the green cloud, within the orange cloud, and these are the neural responses to those different odors. If you look between the clouds, the correlations drop down naturally because the clouds are different. They're separated. It's on the order of 0.58 to 0.82 or something like that, and that's the mean. So it drops between clouds. Now what if we train the bees, one cloud, all the different mixtures in that cloud mean sucrose, all the different mixtures in the other cloud mean nothing? Do we see separation? And we do. So what happens? Suddenly, in trained bees, the correlations within the green cloud go down a little bit, so the cloud gets bigger, gets a little bit more spread out. Same for the orange cloud, but the distance between the clouds increases. So what happens in the bee brain to these more complex mixtures, the clouds get bigger, but they get driven apart. And it helps bees classify. Then saying, if I get something new, it falls within the boundaries of the orange cloud, and I'm going to think it's the same. So it helps with the classification problem. And this is a way I have of representing it. So what happens in the antennal lobes, driven with the plasticity is that you get these clouds of points, and when you teach them that the orange is good and the green is bad, the antennal lobe, the neural network, separates the neural representation. And I won't get into some of the work we've done in the mushroom bodies, is that because those mushroom bodies, this is the information that gets fed into the mushroom bodies. And the job of the mushroom bodies is to say these things are the same thing. So you represent that by saying they make them more invariant. They converge on the same thing. And that's the job of that complicated network that the antennal lobe feeds into. And again, before I get into the last part of my talk, and this is actually the basis for starting some of the work we're just getting going on groups, this is a model that my colleagues at UC San Diego have produced with the antennal lobe and all the cells in the mushroom bodies, including behavior groups, to explain the way all of this plasticity is integrated into making a decision. And again, this is another talk I could give. But this model behaves in terms of making decisions about odors, reinforced or not, is the same, is not, behaves very much in the same way that bees do. And we now have a grant proposal pending where we're going to test some specific predictions of this kind of model. But when I say we feed model bees into a group, they all have a brain that looks like that now because we've got the computational models developed. So let's come back to that other kind of reinforcement before I segue into the group behavior. If you remember, I said it was really important for the bees to learn to ignore things, right? Just like it is for us, just like it is for small roundworms. And several years ago, I began to wonder, this wasn't the only reason we started working on this, but I began to wonder, well, if the bee feeds on sucrose, there's a physical stimulus that says reinforcement. There are sucrose-sensitive taste receptors that feed into the brain. They start firing away. They activate the VUM neurons, and octopamines mean sugar. So I thought, well, when there's an absence of reinforcement, if there's an absence of a physical stimulus, and the bee's learning about that, what kind of reinforcement pathways would be able to represent nothing? And we're able to take advantage of some genetics. This is a big lumbering drone honeybee. Drones are haploid. They grow up from unfertilized eggs. And they grow up into adults. We can take the adults, move them into the laboratory, and they learn perfectly well. So we can train a queen's gametes, literally is what this means. We can train virgin honeybee queens. I'm sorry you can't see the axes here. We can train drones and queens. They learn very well, just like workers. When we train the drones to ignore an odor, and we train the queens to ignore an odor, and they do, they don't learn that odor very well. These are drones and queens. And again, sorry about the axes, but these are just the frequency of proboscis extension that you saw earlier. These are the ones that really learned to ignore the odor, so they have trouble learning it when they're trained to it. There are other drones and queens that, in spite of how many times you hit them in the face with an odor and you subsequently train them, they learn it very well. So we can take these queens and mate them to these drones. We can take these queens and mate them to these drones, and check the workers, and we're going to call these non-inhibitors. So if I say non-inhibitors from here on in, this is what I mean. And these are workers, and this is when they're conditioned to a novel odor, not the pre-exposed odor. They learn it fine. When they're conditioned to the pre-exposed odor that they've been exposed to 40 times, they learn that fine too, right? But we get another genetic line where they learn the novel odor, but they've successfully learned to ignore this odor. So the inter-individual differences that we were seeing coming out of colonies in these learning tasks were individual differences. They were genetic differences. So many people just sort of average the good learners and poor learners into one learning curve, but I became very interested in individuals, and I challenged my very first graduate students to figure out why and I said, I thought it was going to be age because I wanted to write a grant to NIH to study Alzheimer's in bees. And then I said, if it's not that, it's motivational state, behavioral cast, guards, nurses, foragers. And they did a lot of work and proved that it was neither. And then the thing that we had left was genotype. And when we controlled for genotype, we saw this is genetic, these differences. It's also represented in the brain. We've done some work in the antennal lobes now, and this is one odor. This is another odor. And this is the mixture, the 50-50 mixture. These are just the transients I showed you earlier. Now, when we take this odor and we pre-expose it, what happens to the mixture, and this is a little bit light, but the mixture becomes biased towards the novel odor. It's getting subtracted out. So that odor we've trained them to ignore is already getting subtracted out at one of the first levels of processing. So this is an important part of the latent inhibition. It's not all the story, because I told you the story is more complicated, involving a visual context that undoubtedly involves other parts of the brain. But you can see that it's represented in this neural activity, and likewise, you do the same to this odor, and the mixture shifts in the other direction. Modeling, and you might get an idea now that I work with a lot of people who do computational modeling. We could set up our neural network, our model, that mimicked very well this pre-exposure effect that I just showed you. And we started to ask, well, what sorts of changes in that network best produce this effect? And again, you're modifying the inhibition. You're modifying the inhibitory connections in these cells, right? So a general rule that we found is if you want any neural network to learn a pattern, the best way to do it is to target the inhibition. That will change things most reliably and quickly. But now, what is it that's changing? We could do, twice now we've done it's called quantitative trait locus analysis, where we could take our genetic lines and the high line for inhibition and the low, we can cross them, generate hybrid recombinant queens, an F2 queen, and look at drones from that queen. Because remember, the drones are her gametes, and her gametes are going to represent genetic recombination from the high and the low line. So across 500 to 1,000 drones, we can pick out drones that show really good inhibition, very few responses to that pre-exposed odor. We can find drones that behave as though that didn't matter at all, so they learn fine. We've done this twice with these drones, and then you can go through and look at genetic markers. In the most recent case, they were called single nucleotide polymorphisms, where their location in the published honeybee genome is known. And we can ask, well, which of those markers now correlate to one or the other type of behavior? And there are a couple, but one has shown up in all of our analyses. It's at a particular site in the genome and is highly linked to this behavior. So we asked many years ago, this is that EST941 location right here. We asked, well, what's there? You can go in the genome and start asking what is around there. And of course, there are many genes right here in this region of the genome. And first thing I want to point out, and this will be important for the group level behavior we talk about here in a few minutes, is we were doing this at Ohio State University. And independently, Rob Page, who's also here, as you know, when he was at Davis, was selecting for other kinds of foraging traits, pollen versus nectar collection, doing a similar kind of mapping study, and independently they identified the same locus. So this locus comes up in everybody's independent screens multiple times for us. So there's a gene or genes there that are somehow very important for these foraging-related behaviors. So what's there, and one thing that really stood out for us is this one right here, and it's a tyramine receptor, a biogenic amine receptor, right? So tyramine, the biogenic amine, octopamine, biogenic amine, relationship to plasticity. And it turns out that tyramine, the reason it stood out is tyramine and octopamine have been called the invertebrate counterparts to our adrenergic system, adrenaline, noradrenaline. These cells, moreover, in the process of making octopamine, must make tyramine because it's a biosynthetic precursor to octopamine. So they make both tyramine and octopamine and probably release it as cofactors. So we started to wonder with this tyramine receptor, what is it about it? And across individuals, there's no mutation in the gene that we can find. Mutations are all in regions around the gene, so we think it's a regulatory element that's being selected here that differs across individuals. And we were able to test it in our studies, showing that when we can, and this is something that's a little bit hard to understand, as if nothing I said today is hard to understand. You have a gene for a behavior, and you think, okay, I want to go in and knock that gene out. I should knock out the behavior, right? So I expected, here's that line that goes up for the novel odor. Here's the line that goes up for the pre-exposed odor. This line should move up to meet this one, right? That's not what happens. Basically, what that graph shows is the pre-exposure odor goes way down, and it drags the other down to meet it. So what we've just done, by knocking out the gene, we've enhanced the behavior. So what it means is that an intact tyramine receptor is important for modulating the difference between excitation and inhibition. It's very important for saying, what does this experience do? Does it drive a network one way or the other? And it's doing so by the way this tyramine receptor functions. Okay, so now let's get to the new parts of my talk. Now that you know all about reinforcement pathways in the brain, and this is there was a call for proposals at NIH a few years ago called Modeling Social Behavior. And I immediately thought, well, that's certainly something honeybees do better than fruit flies. And maybe they would be interested in this foraging problem. So we wrote a proposal with Jürgen is on it, my colleagues at UCSD, a new postdoc, Chelsea Cook, who's here collecting some of the data. And we wrote a proposal building in all of the modeling and all of the genetics, the decision making in the brain, the plasticity, everything I just told you about. And we argued that we can now study social behavior from the gene through the intermediate neural networks of the brain, how they influence behavior and how that behavior influences group. We can go all these different levels. And they bought it. So now we're just getting started, and I just want to tell you the kinds of things we're starting on. It was based on the hypothesis. First, let me back up. When bees are foraging in a group, you can identify bees that are explorers, the ones that tend to go off and find new things, and exploiters, the ones that actually try to collect based on what the colony knows. And it's important to have both kinds of bees in the colony. So we thought we have these inhibitors. Remember, they're the ones that don't like not getting reinforcement. And we have the non-inhibitors, the genotype that says it's okay to not get reinforcement. And I thought, well, by virtue of the bees' actions when they land on this field of flowers, they're going to deplete the nectar. So pretty soon, what was all good at first, you get some flowers that are depleted. Well, who leaves? They're the inhibitors. And the inhibitors, when they leave, because they don't like this patchy reinforcement, they're going to be off finding the new kinds of flowers that just came into bloom. So that's the basis of we're now starting to call the inhibitors from our laboratory-based analysis the explorers, because they're going to be off finding new things. The non-inhibitors are the exploiters, because they're going to go pick up what's left here. And it's the basis for this division, that we're studying, that was the basis for our proposal. So, like I said, we're proposing now to study these group decisions completely from gene regulation to collective colony behavior. And I just want to show you a little bit now of the modeling that we've done and some of the data that we're collecting. We're going to assume that foragers are a mixed batch of scouts and recruits. Scouts find the new food. Recruits are the ones that come out to exploit what's known, right, based on what I just showed you. The high LI, because of the what we call the high latent inhibitor line, because they're sensitive to the lack of the reward, they make very few flights to the same location. They go back and recruit, and then they're going to be off finding something else. The low LI, they don't mind the repetition, they don't mind the lack of reinforcement, and they keep returning to the same location. This is based, by the way, on what you may know as the honeybee dance recruitment system, right? A honeybee, when it's off exploring and it finds a new really good food source, it returns to the colony, and I should have put a video of the Waggle Dance here, but it performs a series of movements that tell the bees following it the nectar or the pollen or whatever is out in this direction relative to the position of the sun. So if the bee's dancing 45 degrees to the right of the gravity vector vertical, whatever it found is 45 degrees to the right of the present position of the sun. And the length of one part of this dance is correlated to the distance the bees have to fly. So just following this kind of symbolic language, not as rich and diverse as ours, but it is a symbolic representation of sun and distance. The bees know approximately what direction and what distance to fly. So it's a sophisticated recruitment system. So I'm going to show you a couple of graphs just to show, and we're just at the start of these studies, about modulating the proportion of bees who are scouting versus the proportion of bees who are exploiting, and how the persistence has an impact or interacts with some of these proportions. So you can imagine a bee on a foraging flight, and a scout bee will fly out the circuitous route, find some place maybe a mile or two distant, quite some distance for a bee, an animal the size of a bee, and then it turns around and finds something and it makes a beeline, literally, that's where the word comes from, back to the colony, which means it's constantly integrating its rate of turning and speed so that from any point on this complex path it can calculate the approximate direction back to the colony, performs this series, this dance language, or the recruitment dance, and then the recruits will fly out to the resource and begin to exploit it. So we set up some models, and these are just some examples of the models with different resource distributions. And these orange points here are scout bees, and when they find something, they run into something green, they fly back, and they start recruiting these other gray dots here, and these are the bees who are going to be the ones who are exploiting a resource. And we can manipulate in our model something much more easily manipulable in a model than in the field. We can manipulate the quality and distribution of the resources, and we can see how these factors then manipulate the success of bees in harvesting what resources are out there. And these are just agent-based models we've been using. We're using a couple different modeling approaches. So, whoops, went too far. So if we say, okay, there's no persistence. There are no real exploiter bees. Everybody's a scout bee, so the bees tend not to be very persistent in any one location. This doesn't happen in reality, but it does happen in our model. And we look at now what proportion or what's the optimal percentage of scout bees depending on a lot of bees in the colony not being very persistent. And it turns out that 10%, 50%, 90%, the more scout bees you have, the better, right? In a case where there's not going to be any exploitation. The more scout bees, that makes sense because they're going to be off just finding things all the time. And if I don't recruit somebody who's persistent at exploiting what I know, there's going to be another scout right behind me to exploit it. But when there's a little bit of persistence, some bees are persistent in what they know, suddenly these curves shift, and for a certain level of persistence, suddenly it's best for a group to have 50% scouts, right? So the level of scouting really depends on this persistence. And really quickly, one of the things we found is that if you manipulate persistence, so each of these curves, this is a low level of persistence, intermediate, and high level of persistence, and we look at what is the percentage of scouts given these different levels of persistence of the exploiter bees. And if you look at the peak of each of these curves, the percentage of scouts that's optimal, where the group does best in our model in terms of exploiting the resources, however they're distributed, you'll see that the optimal balance here is about 80% scouts where you get low persistence, what I showed you earlier. And the optimal level now decreases as you increase the persistence of the colony, or the fraction of bees that are very persistent. So these are some very nice statistics that have come out of our initial modeling, and this is something what I just showed you, shown from the perspective of recruits. It's a little bit different for if scouts tend to be a little bit persistent. But, and I'm going through fast because I only have a few minutes to go through the next hour's worth of slides here. A model's not any good unless you have data. One of the things Chelsea, who I introduced a few minutes ago, is doing is she's trying to go out now and find out, can we identify bees who are very persistent versus bees who are explorers. And we can start looking at what's the fraction in the colony here. And then we can start manipulating the actual resource distributions to see if the colony now can adapt these fractions. And this is one of her first studies where she can go out in some facilities, nice facilities we have on West Campus. You can put bees on these feeders. So all the bees are coming to this wonderful feeder. And then at some point when they're used to that feeder and you have these bees marked so you know who they are, you put another feeder out about 5 meters away and you wait and see who comes to that feeder, right? So you register the bee. Then the next day you have another feeder out, same group of bees, you put a new feeder out 5 meters away in another direction, who comes? Turns out the bees that show up at these new feeders are many times the same bees. Each of these columns here represent individual bees, and these are fraction of times these bees showed up at the new feeder, and this is 0. What it means is that most of these bees, when they find a feeder, they stick with it, right? They're very persistent. But there are some individual bees who are always the ones showing up at the new feeder day after day after day. So we can actually go in the field now and identify these bees and literally say that these bees are explorers. These bees are exploiters. These are the ones we want to try to manipulate with different resource distributions. These are the data we can feed back into our models and say, well, what should happen when we tweak this or that experimentally in the field? Furthermore, genetically, these are the ones that should have a particular version of that tyramine receptor and show strong latent inhibition. These are the ones that should have a different version of that receptor, right? So we can make predictions the whole way from the group down to the level of genes in the group. So we've only had this grant a few months, so I'm extremely happy with the industriousness of my colleagues and all the stuff we've been able to do. But I want to turn to, in the last few minutes, a last project. And finally, for a group thinking about medicine, I'm coming to epidemiology. And I had a very, very wonderful colleague many years ago, Dhruba Nog, who showed up on my doorstep at Ohio State and said he had just gotten a job. He worked with bees. He'd just gotten a job at a small college. He's doing a lot of teaching, but he wanted to do more research, so he was going to quit his job at the small college and could he work in my lab? I thought, well, yeah. And Dhruba had published a paper in 2002 with another colleague, Scott Kamazine, on how the colony organization affects pathogen transmission. And that was in Journal of Theoretical Biology, and he wanted to test some of the propositions. So he came, and I didn't work on epidemiology, but I think it's just unbelievably cool. Dhruba has moved on to other things. He's now a professor at Colorado State. I'm trying to drag him back into it so we can write another grant and we can start some of this work here. But we published a couple of papers, and he wanted to publish another. I want to tell you a little bit about what we found in these papers. He also wrote a grant with me on some of these ideas that went off to the Ecology and Evolution of Infectious Diseases panel at NSF. And one day my phone rang, and I picked it up, and it was the panel director calling me to tell me they decided to fund the grant, so that was really good news. And he said that this grant was so different from everything else that everybody loved it, and we weren't asking for that much money. I made a mental note, never do that again. So they funded it, and we had a series of studies now. And then I went looking recently, or just the other day, as I was preparing this for the title of our grant, which was on the previous slide, and I actually came up. And I have to put the citation here because this is a slide out of Rita Colwell's talk, who was then the director of the National Science Foundation. She gave a keynote lecture apparently at the Asian Conference on Diarrheal Diseases and Nutrition, where a good part of her talk was about Dhruba's and my work. So I was very proud to find that out. And I didn't know that before. But anyway, it centers around this idea that when a bee comes back to a colony having found something, one of the first things it does is engages in food exchange to get unloaded by bees in the colony. It's called trophallaxis, where one bee will stick out that proboscis that you saw a few minutes ago, regurgitate some of what it just found, the nectar, and it tells the unloading bee whether this is a good source or whether it's not. And it's a very important means of information transmission in the colony. Is this a good thing over here? Is this good relative to something else? Well, the bees in the colony know because they're unloading foragers who are coming back from these different areas. One of the things that Dhruba was proposing was that this trophallaxis can also be involved, this social interaction that's so important for organization of the colony, can be involved in the spread of pathogens and toxins that the bees have been exposed to while they were out collecting flowers. Now, to understand how, we got to tell you just a minute, I'll take a minute to tell you about the guts of a bee. When a bee takes in nectar, or sugar water in our case, it follows through the long esophagus into its abdomen, where the first part of the gut is called the honey stomach or the crop. And it's like a big balloon that distends. And the nectar is stored there until the bee comes back to the colony, and it's from this crop, the honey stomach, that it regurgitates the nectar to feed to other bees. Well, some of the things in the crop will pass to the midgut, where it's absorbed into the bee's hemolymph, its blood. And the regulation of movement has to go through a device called a proventriculus, which is a muscle-activated device for regulating the flow between the crop and the midgut. And it turns out that passage has been well known. Passage of particles like pollen grains, or in our case pathogens, from the crop to the midgut depends on the viscosity of the solution in the crop. More viscous means slower movement. Depends on the size of the particles. Bigger particles tend to get stuck in the proventriculus longer before they get passed. The important part here for a pathogen is that as long as the pathogen resides in the crop and is not passed to the midgut, it's going to remain infectious through trophallaxis until it gets passed through. So that length of time we'll call the infectious period. So Dhruba set up an ingenious experiment where he could create a small colony of bees with 6 different age cohorts that are all marked. And as the bees structure their age cohorts, the oldest ones are going to be the foragers, and they stay on more or less the outside of the social group. The younger ones will be on the inside of the social group. So there's a structure to the colony. The group structure somehow is now correlated to the age distribution. And then we're going to take some of those foragers coming out from these marked social groups, Same picture you saw a minute ago. You can train the bees to come to a feeder, but now, instead of putting actual pathogens in there, which you can do, Druba had this idea of putting these inert polystyrene microspheres. They're almost microscopic, less than the size of a pollen grain. The bees can pick up these microspheres while they're feeding, and they come back to the colony, regurgitate the nectar, the sugar water, with the microspheres, and then afterwards you can look across this social group and pick bees of different ages and ask, do you see the microspheres 4 hours after the return of these foragers? Where do the microspheres show up? So one bee trophallaxes with another, who trophallaxes with a second, trophallaxes with a third, and you can use these microspheres to look at the social groups. And you literally dissect out the crop, burst it open, put them on a hemocytometer, and you can figure out the density of microspheres that are carried by any bee in this chain in the social group. And I want to say that something I remember the reviewers of this proposal really loved was they're inert. The problem if you do this with a pathogen is the pathogen reproduces at every step, right? So because these are inert, we could get clear estimates of the infectious period for these bees. Not confounded by the biology of a pathogen. And just as an aside, you can get these microspheres of different sizes to simulate pathogens of different sizes and how they're going to affect this. Well, Druba could feed bees, and this is the evil tie the bee down experiment, put its face in some sugar water. If you feed them thick sucrose, he was able to show you get longer crop retention, potentially a longer infectious period because it increases the time that the bee can regurgitate the sphere. Thinner sucrose passes through much more quickly, and you get short infectious periods. So this is time since feeding in minutes, and this is the proportion in the crop. And in 40% sucrose, you get much more of the microspheres persisting in the crop than when you have thin sucrose. So we'll call this long infectious period and short infectious period treatments. Well, some of the take-home messages were, if you now look across the bees of different age groups, these infectious periods have a big difference of how this information, these potential pathogens are passed through the social group. Long infectious periods, you see this kind of distribution across the bees in the colony, with many more bees having a lot of pathogens. Short infectious periods, most of the bees, half of the bees you sample, don't have any putative pathogens in them. So this infectious period is very important for how this will move or how any potential pathogen will move through the social group, and I'll be done in just a minute. Anne's giving me this look. And just really quickly, again, I know I said really quickly several times in my talk so far, right? If you look at each age class, the oldest age class, the youngest age class, where you find these particles, the oldest age classes have a lot because they're on the periphery, they're interacting with the foragers. But under short infectious periods, it doesn't pass to the middle of the group, the younger age classes, as effectively as when you get an extended infectious period. So the infectious period here really has a big influence on passage of material through the social group. And actually, Druba called this organizational immunity, depending on the particular infectious period that's being used. And lastly, and I'm not going to talk about this, he went through and could actually define these social networks of bees of different ages, doing some tedious observational studies, finding that long infectious periods tend to have greater degrees of interaction. Individuals who are infectious will interact with many more individuals than with short infectious periods. And it turns out they tend to be much more clustered in a colony. So there are lots of these statistics that he could go through and invent. We could identify bees that were highly infectious. They were super spreaders. So there are certain bees who are much more influential in spread of something through the social group than others. And this is probably the longest talk I've given in a long time. I'm sorry for I'm a little bit over. Let me end it here, and let me thank a lot of people who I've mentioned, who I haven't mentioned, who've been involved in all these studies. And I'm really excited about taking what we know in the bee brain now out to the level of group behavior, both in foraging, and I would dearly, dearly, dearly like to try that NSF panel again on epidemiology. Thank you.
We are over, but I think we could take a few quick questions.
Did I understand you to say, when you were talking about the drones, and when you were talking about the drones and the queens, that learned behavior then had a genetic expression in the next generation?
A learned predisposition, right? So it's a genetic predisposition to learn one way or the other. So whenever I see a bee now who doesn't seem to be learning well, I used to think that's a stupid bee. But now I'm kind of thinking I'm the stupid one because I'm not asking it the question it's good at, right? So that's what I meant by the learning predisposition.
Another question? All right.
There's more than just the explorers and the
Exploiters.
Is there a more diverse classification characterization system
Yes.
like the bees?
Within either of those classes, you probably have bees with different kinds of experience that let them be better at, within their class, better at one thing, then better than another. Also, one of the things we put in the proposal, we don't know for sure, since the regulation of this gene is since the differences in this gene from one individual to the next probably stems from a regulatory region and not from a mutation in the gene. So one of the things we propose is that bees, even though we call them an inhibitor, that if you change the workings of its regulatory region, you could turn it into a non-inhibitor. So we don't know that these genotypes are fixed even right now, and that's one of the things we want to test in the next few years. So that adds a level of complexity that I didn't even get into.
All right, one last, and then we'll thank him.
Is there, you talk about odor, and then you talk about basically a group thing. Is this like an industrial? I mean, can you project this out to be like an industrial application or a military application?
Yes. As a matter of fact, I have an award from the Army right now. And this is the work on mice that we're doing that I didn't talk about at all. Where I and my colleagues there who think similarly have said that everything we've learned about the way bees detect and discriminate odors is very important to build into the training protocols for animals that are used by the Army and the military to detect odors. Because dogs have about a 50% hit rate. It's not really good if it's a landmine, right? You want it to be better, but that's about the best you can get an animal to do right now. And we have a grant from the Army to develop improved training protocols based on these clouds of points I showed you, where the animal's not always trained on one thing, you're training it on multiple things that all mean the same thing, and then the nervous system says, I get it, and it collapses it down to the same meaning. We built it into some of the training protocols for mice, and we have a grant right now from the Army that we're working quite hard on trying to take this information and project it out into that kind of realm.
All right, let's thank our speaker again.
Thank you.